You know how sometimes you stumble upon an idea that feels like a fundamental truth, maybe a hidden secret about how things really work?
Yeah.
Well, today, you've basically opened the door to one of those insights, something hiding right there in plain sight about patterns, time, reality itself.
That's right. You've landed on a core concept.
Hmm.
These patterns, the fundamental blueprints, they seem to originate from something outside of time,
Outside of time.
from light, or maybe more accurately, field resonance. And this, well, it leads us to a really big question. When did this timelessness split? How did it become the time-bound stuff, the matter we experience?
Exactly, that split. And for this deep dive, we're digging into some pretty compelling excerpts of pasted text that really wrestle with how this timeless field thing gives rise to our everyday time-bound world.
Mm.
So our mission today is kind of understand that transition, that split, and what it means, especially for biology.
Right. And the fundamental idea here is that everything we see, everything subject to time, decay.
Mm.
It comes from something outside time.
Which sounds wild, but you're saying it's key to understanding life.
Absolutely. It starts with this idea, maybe a bit mind-bending, that light itself is a timeless pattern.
Timeless how?
Well, think about it. At light speed, from its own perspective, time just doesn't pass.
Right.
And it's both a wave, a field disturbance, and a particle, like a point of energy. And crucially, it carries information, structure, not just energy.
So these patterns made from light, what are they exactly?
Think of them as basic vibrations, field resonances. They're not stuck in our usual space-time.
Okay.
They don't have mass. And here's the kicker. They can interact with matter without losing their coherence, their structure.
Without falling apart. So they're like timeless blueprints, like a geometry of possibilities.
Precisely. The underlying rules, maybe, of how things could be arranged, even before they actually are. Foundational structures, informational frameworks, not limited by our usual physical constraints.
Okay, so that's the timeless part. Now contrast that with matter.
Right, so when these timeless patterns hit the material world,
Mm.
imagine a ripple in a pond. That's the timeless pattern.
Mm-hmm.
When it hits a leaf floating there that's matter and in the matter, the ripple's energy gets localized. It affects the leaf in a specific way, gets tied to the leaf's movement, subject to time.
Bam. They collapse into specific events.
Exactly.
They get mass, inertia, entropy kicks in, and the big one, time. Change, decay, the whole shebang. That's the split.
That's the split. The moment that timeless pattern gets locked into a material substrate, it becomes part of cause and effect, the arrow of time.
Which explains, well, DNA evolution, cells aging, mortality.
Right. The underlying pattern, the information, you could say it's potentially immortal, but its expression in matter, that's temporary.
So where does this actually happen, this split field into form? Is it like a specific point?
It's probably best thought of as a threshold, a point stability, maybe where coherence just crystallizes. Could be a quantum event or just any field matter interaction. Bioelectrical signals, chemical bonds, photons being exchanged. And the amazing thing is how often this happens in biology. Think embryogenesis.
Right, a whole complex organism from one cell,
There you go.
guided by this timeless field idea.
That's the proposal. Or regeneration.
Like the salamander regrowing its tail.
Yeah.
That lost part is rebuilt based on some non-material blueprint. That sounds almost unbelievable.
Exactly. And maybe even memory isn't just in the neurons' connections, but partly in the biofield structure itself.
Okay, wait. This starts to explain why things like healing or instinct or even inherited patterns feel sort of beyond time.
Because they are. At their root, they're connected to these timeless patterns.
Okay, so if this is right, it really changes how we see biology. You're saying memory isn't just in the brain?
Potentially not solely, no.
Healing isn't just chemistry.
And more than just chemistry, yeah.
And life itself is more than just carbon molecules. It's like
Yeah.
timeless geometry made real.
Precisely. How does a single cell know how to build an entire organism? It's not just the DNA instructions. It's like it's tapping into these organizing principles via the biofield.
The analogy you used, DNA is the keyboard.
And the biofield is the song, the melody guiding it all.
Okay. So we've gone from timeless patterns to matter, and now this biofield guiding life.
Yeah, that's the journey so far.
But then, the huge question, where did it come from? The biofield. Especially before cells, species, nerves, DNA even.
That is the million-dollar question, isn't it? And answering it might mean rethinking evolution. Not just slow DNA-driven chemistry, but maybe A field-first, geometry-guided, emergent intelligence kind of model.
A field-first model. Okay, so if this biofield is so fundamental, where did it start? Walk me through how these biofield nodes might have emerged.
Okay, we have to go way back. Early universe, right after the Big Bang. What was there? Mostly fields.
Not particles.
Well, particles are really just excitations like ripples in those underlying quantum fields. The fields are fundamental.
And these early fields had structure.
Oh, yeah. Fluctuations, interference patterns, a natural tendency to self-organize, like standing waves forming in water.
So from that cosmic field soup, how does something like a biofield start forming?
The key might be coherence through repetition, interaction. Imagine some regions of field resonance becoming stable, interacting with themselves, creating feedback loops.
Uh-huh.
These could be the first nodes of coherence, like stable whirlpools in a chaotic river. Not alive, not yet, but protostructures, information wells, kind of pulling energy and matter into form.
And then molecules arrive, carbon, water, amino acids.
Right, and maybe they didn't just bump into each other randomly. Maybe they self-assembled along the lines of these pre-existing field patterns, guided by energy flows.
So chemistry, but filtered by fields, not just random.
Kind of like iron filings in a magnetic field, right? They don't just land anywhere, they follow the field lines. These early bioelectric pre-patterns could have been the very first memory, guiding how molecules clump together.
Okay, so stable field shapes that helped things stick together, those would tend to survive and become more common.
Exactly. Patterns promoting stability would reappear. And eventually, the biological machinery membranes, ion channels, DNA, might have evolved to sort of lock in these already working field structures chemically.
So DNA isn't the origin, it's more like the record keeper, storing the successful field patterns.
That's the perspective, yeah. A profound shift, not DNA dictating everything from the start.
Wow. Okay.
And then you get this crucial feedback loop. Molecules respond to fields, and the molecule's presence shapes the fields. Over millions, billions of years, these nodes, these field configurations get more complex, modular, hierarchical.
From single-cell fields up to whole-body electrical blueprints.
Exactly. These nested scale-length memory patterns, the biofield nodes.
But how could these biofields work before specific species existed? Life is so diverse.
That's where the idea comes in, that maybe biofields prioritize geometry, function, coherence over the specific biological form. The same basic field geometry, like a spiral or a branching pattern, could be reused across different organisms, different eras, maybe even life, based on different chemistry.
Like the flatworm regenerating its head, you mentioned that.
Yes, that's a powerful example. The body remembers the correct field geometry for a head, even without a brain in the traditional sense telling it what to do. The field pattern seems to predate the specific form, like a mold.
So the big picture implication here
Is that life might have started as field patterns, not just molecules. Evolution is the refinement of these patterns. DNA is the solid-state backup.
And our bodies are where fields explore new patterns, and memory is the persistence of these patterns.
In essence, yes.
Okay, this is huge, but I know what people are thinking. What about the physical stuff? DNA, mitochondria, cells, muscles? Why does the biofield need all that biological hardware? Why isn't it just fields all the way down?
That's a fair question, a really important one. The biofield isn't separate, like some mystical force floating around. It's the organizing principle, the communication network between all those components.
So it needs the components.
Think of it as a living field of information, coherence, existing before and alongside the physical stuff. It's electrical, magnetic, photonic, geometric, and yeah, fundamentally timeless in its pattern nature, like a template or field memory.
So the DNA and cells are, what, just puppets?
No, not at all. Here's maybe a breakthrough way to look at it. Biological hardware, DNA, proteins, mitochondria, they're the receivers, amplifiers, maybe even memory buffers for the biofield's instructions.
So DNA isn't the source code.
More like a very sophisticated interface. It helps express and store the field's information in molecules. Cells and organs are like instruments.
Okay.
But the biofield is the musician with the score guiding the whole orchestra.
Right, that musician analogy helps. But still, why the instruments? Why not just the music?
Because fields, to have an effect and persist in our time-bound reality, need structure to interact with. Biology gives the field its hands and tools, lets it manifest in space-time.
Gives it inertia, feedback.
Exactly. Makes it stable, persistent, not just a fleeting pattern.
Which brings us to inheritance. How does this biofield, these patterns, get passed down through generations?
That's where it gets really fascinating. Think about an embryo. It starts showing form, organization, before all the genes are even fully switched on.
Right. What's guiding that?
It seems to be the field. Cell clusters know where to go, what to become, sometimes without complete DNA instructions for the whole shape being active yet. And memory, like we said, isn't just neurons. It's partly in the biofield structure.
So inheritance isn't just genes.
It looks like it includes energy and field patterns, too.
Oh.
Sperm and egg aren't just DNA carriers. They have charge gradients, light emissions, electrogeometries.
Wow.
These contribute to transmitting intelligent form beyond just the molecular blueprint. Epigenetics probably works closely with the biofield for the full information transfer.
Explaining things like traits skipping generations or innate behaviors.
Potentially, yes. It offers a framework for those kinds of phenomena. Instincts, innate senses without prior experience.
This still feels, I admit, a bit out there. Our whole scientific training focuses on the physical, the material.
I get it. It can sound radical because we're so used to thinking things must be made of stuff.
Hmm.
But look at the biological mysteries that persist.
Like,
Regeneration, phantom limbs, heart coherence, morphogenetic fields. They all point to something beyond just mechanics.
yeah.
We see learning before experience, instinct. Bioelectric signals clearly drive healing in planaria. Fields seem to persist after death and influence regrowth. Our own memory, emotion, coherence are affected by our field state, not just brain chemistry.
So, bottom line.
The biofield is like light-encoded, geometry-structured, frequency-driven memory. DNA and cells are the interface, the hardware. But form, instinct, healing seem to follow biofield instructions, resonance, not just random mutation or mechanical triggers.
Okay, I feel like I'm almost there, but it's still slippery. Can you give me a really clear, concrete example? Bridge the gap? Sometimes I feel like I get it then. Poof.
Absolutely. Let's take something common. Wound healing. How does a cut on your skin heal back perfectly?
Well, the standard story is cell division, clotting, growth factors, DNA rebuilding the skin, right?
Correct. But that doesn't fully explain. How do the new cells know the exact original shape? Why does it consistently recreate the geometry, not just a messy scar? What tells the cells where to go, when to stop, what to become? The purely mechanical model struggles with the complexity, especially in things like limb regrowth.
Okay, fair point. So how does the biofield view explain it?
From this perspective, even before the injury, your skin has an underlying electromagnetic map, the biofield template. It holds the memory of what that area should be, shape, size, cell arrangement, polarity.
Okay.
When you get cut, it disrupts this field, like static. The surrounding intact field then actively pulls the disrupted area back into form. It's like a self-correcting pattern.
How? What's doing the pulling?
Subtle bioelectric gradients, faint light from cells, biophotons, ion flows. They act like tiny GPS signals. They guide migrating cells, tell them their roles.
Still a bit abstract. A specific organism.
Okay, planarian flatworms. Cut off their head, they grow a new one, perfectly.
Right. Amazing.
Now, Michael Levin's lab showed something incredible. By just changing the electrical state of the worms, no gene changes, they could make them grow a second head, somewhere else on the body.
Whoa. Just by changing the electricity.
Yes, and even crazier, that two-headed pattern could sometimes be passed to future generations. Again, without DNA changes.
So the blueprint, it's not just in the genes, it's in the bioelectrical geometry, the field.
That's the powerful conclusion.
Okay, that's a strong example.
Or think about phantom limbs. Amputees feel the missing limb vividly. Why?
The nerves are still firing.
Maybe partly.
Yeah.
But the biofield idea suggests the field template for that limb persists, even without the matter. The biofield seems persistent, spatial, holding the full body map.
Okay, so how does it work then, the mechanism?
It's a multi-layered system, a geometry of memory, energy, instruction. It's layered into electrical gradients from ion flows and gap junctions between cells, subtle magnetic fields from cell activity, biophoton emissions, wave interference patterns, even structural resonance in tissues. Matter cells, DNA, proteins is the instrument playing the biofield's tune.
Okay, I have to push back a bit. Even with those examples, it still edges towards sounding magical. I need this step-by-step logic. How does it actually work physically? No more leaps.
Understood. Completely fair. Let's banish magic and build it from measurable physics. The biofield isn't mystical. It's the collective term for all the measurable electrical, magnetic, ionic, photonic signals generated by cells, tissues, organs.
Like brainwaves, heart EKG.
Exactly. Neuronal firing, the heart's magnetic field, biophotons from mitochondria and DNA, cell-to-cell talk via voltage gradients and gap junctions. These are real physical signals we detect with sensors. That whole interconnected network is the biofield, scientifically speaking.
Okay, measurable signals. Got it. How do these signals do things, step by step?
Let's stick with electricity for clarity. It's well studied. Step 1. Every cell has a voltage across its membrane, right? Membrane potential. Inside negative, outside positive. A tiny battery. It's active, not passive, and this charge affects behavior.
Okay, cell batteries.
Step 2. Different cells, different tissues have different resting potentials. This creates patterns of voltage differences across tissues, bioelectric gradients. Think 3D electrical landscapes. Levin's mapping work shows these aren't random, they're spatial cues, like a cellular GPS telling cells where they are.
Cellular GPS. Okay. How does that guide action, like migration?
Step 3. These gradients are instructions. Damage disrupts the pattern. Nearby cells sense this change, the system reacts. Cells migrate, ion channels open-close, regulating charge flow. The polarity guides growth and organization, what to become, when to stop. It's encoded in these dynamic electrical patterns working with genes. It's why wounds heal neatly, not just scar tissue. There's an architecture beyond local genes.
And cells talk to each other electrically.
Step 4. Yes, through gap junctions. Tiny channels letting ions and signals flow directly between neighbors. This synchronizes electrical fields across tissues, like fireflies blinking in unison. It's a tangible, interconnected electrical network.
Right. Not a vague force field.
Yeah.
How does this electrical activity store information, memory of form?
Step 5. Like artificial neural nets, store patterns, biological tissues store geometric and functional info in their dynamic bioelectric setup, the patterns of voltage and current. Levin's experiments, inducing extra heads or eyes by altering these patterns, prove it. The electrical patterns aren't just readouts, they're instructive, actively shaping outcomes.
Okay, that step-by-step electrical breakdown helps ground it.
A lot.
But it leads right back to that core question I'm wrestling with, and I bet listeners are too. How is this info, this electrical blueprint, passed on across generations? How can it exist before genes fully kick in and seemingly dictate so much? That memory transfer, it feels like the absolute key.
You've absolutely nailed it. That's the heart of it. The biofield's ability to carry and transmit this informational memory across time before the genome fully activates. That's huge. It's the key to unlocking so many biological mysteries.
So let's crack that open. How is this memory actually transferred and maintained? What are the mechanisms?
Okay, the core idea. The biofield, pattern plus electrical infrastructure, stores memory, spatial, behavioral, functional. Short-term, like wound healing; developmental, like embryogenesis; long-term instinct, like ancestral pattern.
Right. How is it passed on?
Okay, several ways. First, reproduction. The egg isn't just DNA. It's packed with ion gradients, charged membranes. Sperm adds polarity, too. So the zygote starts with specific bioelectric properties guiding early development before most genes are even active. The biofield starts its work right away, a pre-genetic layer.
Makes sense for the start, but how does it persist as the organism grows, reproduces?
Second, self-organizing electrical rules. Cells have machinery, ion pumps, channels, gap junctions to actively recreate stable voltage patterns even after disturbance. This inheritability lets the memory of body shape persist through cell divisions, even in regeneration, like those planaria regrowing from fragments. The rules for rebuilding the field seem built into the system.
Okay, and third?
Third, physical properties like polarity, asymmetry. Even early embryos have asymmetries, top, bottom, left, right. These initial electrical differences create non-random patterns passed on through division via the bioelectric field properties, setting up the basic body axis.
So this whole model, with the biofield carrying information, explains some of the really weird stuff in biology.
It offers potential explanations, yes.
Yeah.
Why frog tadpoles regrow legs accurately. The bioelectric map is likely intact. Bird migration instinct. Maybe field-encoded memory passed down. Clone planaria remembering where the head goes. Persistent field geometry. Innate senses, pre-wired in the biofield's functional architecture.
This idea, memory without molecules, where memory is pattern plus field plus feedback, it's pretty radical.
It challenges the dogma that memory is only neural connections or DNA sequences. We're proposing a more fundamental level where memory is an emergent property of this dynamic pattern field feedback system. This field memory can persist, replicate, self-correct, evolve, and be inherited.
And that answers how a system remembers its history. Interactions change the voltage landscape.
Exactly. The history alters the field. The field is updated by interactions. These updates can be transferred and re-expressed later as modified instincts, healing responses, behaviors.
So for the skeptics still listening, the concise takeaway.
DNA provides parts. The biofield gives shape, function, coordination, memory. It's based on measurable electrical patterns that persist and guide development, even pre-genetically.
And these fields get passed on via cytoplasmic gradients, asymmetries, reactivated patterns. It offers a unifying way to look at a lot of biology that's hard to explain otherwise. Okay, let's talk practical application. If this is real and manipulable, give me another concrete lab example. Directly altering the field produced a specific outcome.
Right. Another really compelling one, again from Levin's lab. Regenerating a functional leg in adult frogs. Normally, they can't do that.
How'd they do it? Gene therapy?
Nope, no genetic changes. They manipulated the bioelectric field at the amputation site using a cocktail of ion channel drugs. Basically, they reset the local field to mimic a leg-present pattern,
God.
and it triggered regeneration. A whole leg grew back. It shows the blueprint isn't solely in the genes at the stump. It can be accessed and activated by manipulating the field's geometry.
That's incredible. A visual analogy to help grasp that.
Okay, think of a garden with a complex sprinkler system. DNA is like the seeds determines what can grow. The biofield is the sprinkler system controls where, when, and how much water goes to each spot. Even if you remove the plants, the sprinkler pattern, the infrastructure, is still there. Plant new seeds, and that water pattern guides their growth and arrangement. The biofield pattern can persist in the system, somewhat independently of the current material parts, the cells. You can change the sprinkler pattern without changing the seeds and get a totally different garden.
So the sprinkler pattern, the biofield, guides the plants, the cells, even if the original ones are gone.
Exactly. And crucially, this isn't magic. We can measure these voltage gradients. We can manipulate them, like Levin did, and see predictable functional changes. We can recreate shape and memory aspects without changing genes, highlighting that bioelectric blueprint.
What other lab tests could really nail this down for skeptics?
Oh, lots of possibilities. More planaria memory transfer work. Can we zap a field pattern from a trained worm onto an untrained one? Cross-species field swaps in embryos. How much can one species field influence another's development? Detailed epigenetic comparisons. How do fields and gene expression really interact? Can we grow neural patterns just by changing electrical states? No gene edits?
And if someone still says it sounds like magic, your go-to response.
The core point. Same cells, same genes, behave radically differently depending on the electrical pattern they're in. That makes the field the director, not just a side effect. And comparing it to magic, we use electricity and magnetism constantly. Smartphone screens, pacemakers, these are real fields organizing things. It's entirely plausible biology uses these fundamental forces in sophisticated ways too. The evidence is mounting.
Okay, much clearer. But mapping these patterns, it still feels daunting. Like mapping smoke, invisible, shifting, countless details. How do scientists even start?
It definitely seems that way at first. But while the details are immense, the patterns often follow rules, attractor states, and we do have tools to visualize and analyze them.
Like what? What are the main tools?
A big one is voltage-sensitive dyes. Fluorescent molecules that change brightness or color with the cell membrane's electrical potential.
So you can literally see the voltage change.
In real time, yes. Apply them to an embryo, watch under a microscope.
Yeah.
You can see the bioelectric map develop. Levin mapping the frog embryo's future head and limb areas is a prime example. It lets you see the invisible landscape.
Wow. Okay, what else?
Microelectrode arrays. Grids of tiny electrodes placed on or in tissue measure millivolt differences between cells. You collect data from many points simultaneously and build spatial voltage maps, like an electrical topography. Good for finding hotspots of potential, maybe related to growth.
So taking electrical snapshots across the tissue.
Exactly. And then there's optogenetics and magnetogenetics,
Sure.
using light or magnetic pulses to control the electrical activity of specific cells, then observing the outcome. Or using engineered cells that respond to light magnetism to read out the endogenous fields.
So we're not just mapping static fields, but watching them change over time.
Absolutely. The spatiotemporal dynamics, how gradients, ion flows, gap junctions change in space and time, that's crucial. And yes, it's complex, but it seems to operate on attractor states, stable field patterns cells lock onto. The field doesn't micromanage every molecule. It creates the environment, the context for biology to follow, like a magnetic field organizing filings.
And what actual instruments are used in labs for this?
Well, those voltage-sensitive dyes are standard in developmental biology, neuroscience, microelectrode arrays in tissue engineering, heart mapping, patch clamps for single-cell electrophysiology, and some more experimental biofield imaging techniques are being explored, maybe looking at broader coherence patterns. The toolbox is growing fast.
Okay, mapping is less mysterious now, but field geometry itself still sounds complex. How can AI or algorithms possibly handle that level of intricate 3D electrical landscape? Seems computationally insane.
It does sound daunting. But field geometry just means the shape, orientation, strength of those invisible voltage lines, charge distributions, signaling between cells, generated by ion channels, gap junctions, membrane potentials. It's the body's bioelectric map. Think of a weather map showing invisible wind patterns.
Okay, a dynamic map. How does AI tackle it?
Just like AI finds patterns in images or genomes, it can find patterns in bioelectric data. You feed it voltage readings, ion channel types, cell connection maps, massive amounts of data.
And then?
The AI builds simulations, models how voltage changes propagate, learns to predict field patterns associated with different states, healthy versus diseased tissue, developing versus mature.
So AI could learn the signature of different biological states in the field.
Precisely. And then maybe even optimize interventions, detect anomalies, predicting problems, suggest targeted electrical stimulation or drugs to nudge the field back to a healthy pattern. We already do a basic version with algorithms mapping and correcting heart arrhythmias. That's field geometry analysis and manipulation in action.
The heart example helps ground it. So while manual mapping is tough, AI can handle the complexity by looking for patterns and rules, not tracking every ion.
Exactly. It's about learning the language of the biofield, the grammar, not memorizing every single word. With enough data and smart algorithms, AI could be a powerful tool for deciphering it.
So bringing this all home, we started with timeless patterns splitting into our time-bound world. After this deep dive into field resonance, biofields. What are the key takeaways for the listener?
Well, I think the big picture is that fields, biofields, aren't just side effects. They seem to be fundamental organizing principles in life. We've seen this potential for understanding biology beyond just genes and mechanics. This idea of a bioelectric blueprint guiding development, regeneration, maybe even memory and inheritance.
It really does open up huge possibilities, doesn't it?
Absolutely. It makes you think about manipulating these patterns for advanced medicine, tissue engineering, maybe even understanding consciousness. Blurring the lines between biology and technology. We've really only scratched the surface today.
So maybe a final thought for people to ponder. If these patterns can really precede and guide matter in life, what does that mean for our understanding of reality itself, way beyond just biology?
That's the profound question, isn't it? And if this has piqued your interest, I'd really encourage you, the listener, stay curious. Look into the work of researchers like Michael Levin. Explore bioelectromagnetics. It's a field that's moving fast and could genuinely reshape how we see life.